Good morning, one and all. First of all, I would like to thank Dr. Sumit and his team for giving me this opportunity to talk about a subject which is very close to heart. So my topic given to me is points to consider while developing diagnostic or point of care techniques. But I will be focusing more on point of care techniques and more so ever the expertise which we have, that is lateral flow immunoassay. So before I begin with the actual topic, that is the points to consider, let's just brush through the basics of point of care tests. So what is a point of care test? It's a laboratory testing that takes place at or near the site where the patient is located. That absolutely does not mean this, but we definitely know now the market is high. There is a competitive and very meaning everyone wants to develop a point of care test in each and every disease, disorder, or analyze. And I think we have to give it to the COVID times to start this, because I think during that time we all realized that we as academic institutes, private companies, all of us can develop the point of care technique in India itself, making it very valuable to our patients. So as I said, coming to the definition of point of care test, it is supposed to take place at or near the site of where the patient is located. The College of American Pathologists added 2 more points. That is, it should not require any permanent dedicated space, and it could be performed anywhere outside the physical facilities of a clinical laboratory. The POC, mainly people think it's like a pregnancy test and that's it. No, it can be the simplest dipstick, and it can even range to sophisticated automated molecular tests, as long as it's portable. That is, your analyzers and imaging test needs to be portable. So it can be defined as POC test. The point of care test can have many synonyms. We all know this. They are called as near patient testing, ancillary testing, bedside testing, etc. And we all know the n number of point of care tests which are there in the market. All of us have someone in our family using the glucometer. And during the COVID time setting, we all have used the COVID antigen and antibody kits. So WHO came with a fantastic assured criteria. So if you are developing a point of care test, you should see that it falls in this criteria. So A for affordable, S for sensitive, S for specific, U for user-friendly, R for robust and rapid. That is, the test result should be at least within 30 minutes of time. E, equipment-free, and D, deliverable to those who need the test. So this is one point you should keep in mind when you're developing. Now, we know that there are many advantages of your point of care test. It improves the quality of patient care. You can give an immediate intervention or treatment, which is a plus point of care test. Then the best part of it is the speed. That is the less turnaround time for the test results, and the smaller sample volume which is required. Now, everyone should know the disadvantage, and the main disadvantage is anyone will be performing your point of care test. So this could be the caregivers who are actually not laboratory people, then your less skilled personnel, so there is a chance of error and accuracy. So this device is very important when you're developing a point of care. Remember, the device which you make today may not be of any importance tomorrow. We have seen this with many COVID antibodies. Not only that, since the market is very high, any company which comes out with a more sensitive and specific and cheaper, your kit is going to go for a toss. So it is a highly competitive and rapidly evolving market. So we know this is what happened before. We used to go to the doctor, then they tell us to do tests, then we have to again take a reappointment. But now with the point of care testing, you get the results immediately and the treatment would be given in one short visit. Now, coming to the ideal requirements, I focus on ideal because we cannot actually go ally to each and every point of this. For example, one of the ideal requirements is the first result should come in 1 minute or less. This is not possible in all point of care tests. Now, very important is if you have your quantitative assays, so it should be portable instruments and portable cartridges. It has to have a single simple operating protocol. The most important is you want to do this on non-processed samples. That is like whole blood, urine. You don't want any centrifugation steps prior. Then it should have a built-in integrated calibration and quality control, and the ambient temperature storage for reagents. Should I go ahead?
Just a minute.
Yes. So then going ahead, you need to have a low instrument cost, and the results need to be provided in a hard copy or should be transmitted. So now, your POCT could be rapid, it could be a benchtop, transportable, disposable, portable, or handheld. And you have different types of point of care tests. Now, this is the basic point of care test which I'm going to brush through, but we know that right now you even have molecular rapid tests which are coming in the market, your LAMP. So these are all new, but the very well known are classified into two types. You have the single use POCTs, which could be qualitative or quantitative. And then you have the multiple use benchtop devices. So when you come to these basic point of care tests, they basically run on four principles. One is your reflectance. The urine dipstick is an example of this. Then you have your lateral flow immunoassays, your COVID antigen and antibody kits. Electrochemistry is basically the principle of all benchtop devices. And your electrical impedance, an example of this is your complete blood count. So coming to the single-use qualitative strip, this could be a simple dipstick or a complex strip or an immunosensor where you have a tagged antibody. Whereas your quantitative test, basically these are also strips, but you need a monitoring device. An example of this is your glucometer. So you have n number of strips, which is only a one-time use, but you insert it into a device so that it gives you a quantitative reading. Similar to this is in your anticoagulation, you do your PTINR, you have a POCT for this too. Blood gas analyzers, all this come in that. And then you have the multi-use benchtop. These are mainly point of care tests which are used in your critical care, your ICU, surgical unit, emergency room. So this is where you need the analysis immediately for intervention. So in these benchtop devices, basically you have this pack in these benchtop devices, which lets you analyze a certain number of samples. And after that, you have to replace this pack. But it has all the qualities of a point of care. It is portable. It can be taken to room, and basically you get the results immediately. Then nowadays, you even have these non-invasive. We all wear the sports watch showing our oxygen levels and etc. We use the oximeter. So this is another concept of point of care test. That is the non-invasive point of care. You have the gluco watch nowadays. Over here, what happens is there is a low electric current which pulls the glucose through the skin. And there's an electrode to give you the actual glucose measurement. So these are the non-invasive. Now, after brushing up, I come to the exact point. That is the point to consider when you are planning to develop a diagnostic or point of care technique. Now, the most important point to remember is the need. Is there a need for developing the POC which you're planning to? Why develop it? So now, if it is a disease or a disorder or an infection, you need to assess the prevalence in India and worldwide. And are the cases being underdiagnosed? When it is a particular analyte, you need to know whether this analyte is so important that you need the immediate test results so that intervention could be given either in the ICU, emergency, or at home. Now, the most important part which you will be asked when you submit the project is what is happening to the gold standard techniques. So we all know there are gold standard tests already there for majority of the disease. So what happens is, what is happening to these gold standard techniques? Are they too costly? Do you need technical expertise? Do they take too much time? Are they not available in all areas in India? Which is leading to underdiagnosis. Is your POCT going to be more cheaper than the gold standard test? Most important, you're increasing the number of diagnoses. Do you have a plan for intervention and treatment? Diagnosis and treatment should go hand in hand. So if you're telling you're going to develop, you need to partner up with some pharma or NGO which are going to provide the treatment. There has to be a treatment plan along with it. Now, with all this, you need to have that there is a necessity for quality assurance and the problems with data management. When I say that, for example, COVID antigen kit was for all of us. We know so many people who did it at home and never reported it to the government. Despite of being COVID antigen, they roam freely. So these are the data management problems you are going to face, and whether you are okay with this, to let it be a home kit. So all these things need to be kept in mind. Now, from yesterday, we've been hearing that whenever you submit a project, you have to talk about novelty. But when it comes to technology, should you only develop a technology which is novel and patentable? The answer is no. Right now, a major thing which is happening is the Make in India initiative. The main part of this is you have your foreign companies producing the kits, but they can give any amount, that is, the kit can be costed, meaning can be budgeted for any amount, and we have no control of it. But if you make the same product in India, especially ICMR institutes of government, we can keep a cap of the cost of the kit. So that is why, coming to the Make in India initiative, it does not have to be patentable. You can even do it when it is not a novel kit. Now, when you're developing the point of care, you need to know who is going to use this kit. So if you're making a point of care test where a lay person, like anyone at home, community pharmacists, are going to use, you need to make a kit which is as simple as a pregnancy kit or your COVID kit. If it is for secondary and tertiary care, that is in the emergency room and operating room, then over here, a little proper training and skilled would be required for doing these tests. So this needs to be taken in hand when you are considering. Now I will brush through our institute's experience with development of point of care tests. We have mainly focused on lateral flow immunoassays. So coming to what the points that the points to remember when you're developing. So we first developed a point of care rapid card test for hemophilia and von Willebrand disease. These are bleeding disorders wherein the patients bleed profusely and they die at an early stage because they have the absence of factor. Now, when you come to the main point, what was the need of this? The need is that we are the second largest population again in hemophilia, and if you see our registry, only 1/5 of the patients are diagnosed. So there is an underdiagnosis which is happening. Coming to the gold standard test, what is wrong with them? They are around 3,000 to 5,000 rupees in cost, and you need a coagulation lab. There are very specific instruments which you need, the coagulometer, automated. You need technical expertise. So in all of India, we have a very few coagulation labs who do all the tests for bleeding disorders. So this was warranted that we needed a point of care test in bleeding disorder. And this was endorsed by National Health Mission. And when I said your diagnosis and treatment needs to go hand in hand, they endorsed it. And they said the more the diagnosis, the more they will provide financial assistance to give the factors. Not only then, we even tied up with World Federation of Hemophilia, who said the same. They want to take this kit and go to Africa, where again there is an underdiagnosis. So there too, they would provide as many factors required for these patients. That is the medicine. So this has got Indian and international patent, and this was ICMR funded. Along with that, we have developed a point of care kit for a platelet disorder, again, a bleeding disorder, wherein the patients tend to have a very short life. The same reasons, that is the need. Then ICMR G6PD qualitative diagnostic kit. Now, this is not patentable. This we've just done for a cheap kit. It comes to 20 rupees, make in India product. Then we even have made the COVID-19 rapid antibody detection kit. So now coming to the basics of an LFI. So if you are a technology person and you are into developing technology, the actual principle of an LFI is very beautiful. It's not a simple strip which you see. The principle, if you know what goes in it, it's a very beautiful way of, standardizing something. So when it comes to an LFI, it basically has two unique principles. It has your chromatography and it has your antibody, which are labeled. So it is not an easy job at all standardizing an LFI. People think it is easy, it is not. 10 to 20 things, that is, aspects need to align all together to get a successful kit. So this starts with your antibodies, that is choosing the to get that one pair of perfect antibody which gives you results, you need to do n number of experiments with 10 to 15. So then your LFI membranes, okay, here also you have 4 different components, the sample pad, conjugate pad. They have to be assessed. Each one has its own pros and cons, and you have to do this n number of combinations. Then now for the marker, we use gold nanoparticles, but there are n number of other. Now gold nanoparticles also, if you're developing it, you do get it in Sigma and you have other products which are there. But our main aim is to keep it cost effective. So you need to have a protocol in lab to make your own gold nanoparticles. And then comes the conjugation protocol. So I'm not going to go into this deep, but yes, each and every membrane needs to be tweeted and there are different pore size, there are different sample pads. Each thing has to, for each and every test, you need to do n number of combinations for it. Now coming to the gold nanoparticles, this is also a very beautiful process. It starts with a colorless solution of gold, and then you're just baking, the color changes to a dark black and then a wine color, and you just want that beautiful wine color to come. And even there, depending on the color which you get, it defines your size of the gold nanoparticles, which is also very important. So all these things take place while developing a lateral flow immunoassay. Now coming to the conjugation. You would think just passive absorption is so easy, just put the antibody to the gold nanoparticles and it would bind. That is not true. You can see at different pHs, there's just precipitation taking place. So here too, you have to try with different pHs to get the perfect conjugation. Perfect conjugation is defined when that color of wine deepens and not precipitates or any of that stuff. Now, again, if anyone has performed the ELISA or knows the ELISA, the concepts are the same in LFI. So you have a sandwich format similar to a sandwich ELISA, where you have 2 antibodies against the same antigen, just different epitope. If you get a sandwich, you get a color in the test line. If you do not get a sandwich, that is depending whether the sample has the analyte or not, you do not get a test line. In your competitive, again, you have the competitive ELISA, you have the competitive LFI. Over here, you already stick your analyte to one of the antibodies. Thus, you get opposite results. If your sample has the analyte, then you're not going to get a test line because there's already something stuck to the antibody, and vice versa. Then you have the multiplex. This could be sandwich or it could be competitive. Main part is you want to detect 3 to 4 analytes in one blood sample or one sample, let it be any. Now, as I said, you can use n number of other markers. That is, we use monoclonal antibodies, polyclonal antibodies. We use enzymes for the G6PD qualitative, but you have aptamers also, which you can try out. Similarly, for gold nanoparticles, you have n number of other labels which are there. So your POC could have any one of them. Now, I'm not going to go to the advantages. We all know the advantages of POC. The disadvantage, when you are developing, you should know that an LFI is never going to be quantitative. It's going to be qualitative or it's going to be semi-quantitative. And the most important thing which people do not get is the reproducibility varies from batch to batch. And this is where everything goes for a toss in the market. So ideally, if for a private company, they need to, each batch they produce, they need to validate it. And this is what glitched off in time. During COVID also, the first 2 batches were amazing. And later on, because no validation was taking place, the sensitivity and specificity decreased. So it has to be validated at each and every point. Now coming to developing a POC in an academic institute. These are the instruments which you require. So pH meter, heater, shaker, and incubator, I think each and every lab has. Now what you need for an LFI is the antibody line printer and an LFI strip cutter. Along with this, as I said, you need to order all the antibodies, the cassettes. Now I want to highlight one part, that is you need dedicated staff, at least 2. If you want a technology to be developed, you can't have a staff working on 3 other things and then doing this. You need 2 dedicated staff if you want it developed within a certain period of time, say 1 year. Now, this is how it looks. It doesn't look all grand in your academic institute. So you do it in petri plate. You do it in a very cheap way. That is, you do not draw lines. You use the pipette and you put dots. As simple as that, so that the antibodies are not wasted. When you get good results over here, you move to the next step, which is the line. That also, it looks like this. So everything, we cut the strips. It's like a China market where you cut it yourself, you stick it yourself. Everything is done manually. And once this is in, then you try making it in cassettes. And over here at this point, you do your internal validation, external validation. You can see the difference of how a beautiful kit, by a company looks like and how an academic kit looks like, but that is fine. In the end, we are going to give it to a company who would be beautifying everything. So we have this. So this is the antibody printer, where this is a machine which prints those beautiful lines parallelly. And we have the line cutter, which makes beautiful strips at the end. So this is done at the last stage when everything is standardized. So then you pack it up and you give it for internal validation and external validation. Now coming to the samples, very important. If you have decided to develop a technology, you should be having the gold standard methods along with it. Each and every sample you're going to use for standardization has to be first run with the gold standard method and then only used. Simultaneously, while internal validation, you have a sample size calculation depending on the prevalence. You do it with all samples first with the gold standard, then your point of care test. Similarly, for external validation, over here in external validation, you get the opportunity to choose your institutes where you want to send your POCs. Choose the laboratories which perform the gold standard tests day in, day out. And this could be given nationally, generally it's given nationally, but for our hemophilia kit, since it was even warranted outside, we had given it even to international institutes for validation. So along with CMC Vellore, AIMS, KM Hospital, where you know these tests are being done, we had given it to two companies, that is Steve Kitchen's Laboratory and ISTS. So basically we do the QA/QC of our lab with them. So it was easy to do this validation with them. So a very important part of the report needs to have your gold standard test, your POC test, the inference, whether they do mass or not, and then calculate the sensitivity, specificity, and accuracy. And any other. So when you give it internationally, they write all words. As I said, it doesn't look beautiful. So they'll say you can see the background, the kid doesn't look beautiful, but it's that all you can ignore, because what they don't get is we are not a manufacturing department where we are going to manufacture. In the end, it's going to go to a company. Another part of validation that no one thinks of is after you transfer your product to a company. Generally, DCGI assigns a laboratory to validate each and every batch they come out with. And generally, it is the manufacturer that is what we've seen in ours. So since we invented the hemophilia and DWD kit, they chose us to do the validation for each and every batch. So as that, you should be ready with samples for doing this validation at a very short period of time. Similarly, for COVID, we all know all the companies gave it to NID for doing their validation. Now, once you standardize everything, a very important part is the instruction manual. It has to be the simplest of simple. Simple, clear, there has to be figures, there has to be limitations. When do you expect false positive and negative? It should mention who should be testing, when you should be testing, what treatments are going to affect. So as the funny figure of the KB standard B is given, it should be as simple as that for someone to do it. It should not be complicated. Another aspect to know is your POCT is highly unstable in different environments. Heat, light, moisture, all of this affects your POCT. So this should not be delayed, however, while your internal validation and external validation, everything is taking place, simultaneously do your stability and storage test. That is, you will make 5 packs, you will keep one pack at 4 degrees, one at 24 or room temperature, the other one at 37 and one at 45. Now, for a month, every week you remove one kit and you check with your known samples, negative and positive. And then it's a month. So you're seeing the consistency of results and for how much time your test is going to be stable at different temperatures. Not only this, it is always advised to go to different settings. So do the POCs at a very cold climate, at a very hot climate, and see what they are, what do they say, whether it works. For example, now our D6PD kit, we saw that in very cold environment, it does not work. We've given a time frame of within 15 minutes, you'll see the results. No, it is after 30 minutes you see. That is but obvious because the enzyme is active only at 30 seconds. This is something which we did not think of. So this has to be put in an incubator for 10 minutes and then run in such cold environments. So that is why the need of doing such studies too. Now, timelines are very important. You should have a timeline. Otherwise, nothing will be done within a set of time. I am highlighting procuring reagents because I feel that is the thing which takes the maximum time, especially with our government institutes through the GEM. It takes 3 weeks, and once the company gets an order, it at least takes 3 to 4 weeks. And when you're ordering for this, you will order very minute volumes initially because you don't know which antibody is going to work. By the time it works and you realize, "Gee, this is the one which works," again, that's 6 weeks protocol start. So I think the most annoying part of developing something is the procuring of reagents. So now, since we have developed, we have faced many problems while developing after transferring it to the companies. So these are the few of the lessons that we learned, and we know that we're going to learn many more. So one thing is never standardize a POCT with one set of reagents. I'll tell you a bad luck over here. So we standardized the kit and it was specific, sensitive, everything was good. Once you do that, the ICMR headquarters, they float the expression of interest, it goes to the company. That easily takes 1 month. After 1 month, when the company got and the MOU is signed, you're supposed to give the technology to them. We gave the technology, they started procuring the reagents. At that point, they came to know the antibody which we use is no more in the market. So what a tragic thing, you know, it's like we thought everything is fine, but that very antibody which gave you specific affinity is no more in the market. They stopped producing it. So that is where we have learned that always standardize it with 2 sets of reagents. So if something happens to one, you have a backup of the other. Second thing is know your disease or disorder or your gold standard test very well. So for a particular infection or disease, if the gold standard tests are ELISA based or flow based, you're going to get your antibodies very easily and cost effective. But if you have diseases where the gold standard tests have nothing to do with your antibody, you're going to get a hard time searching them in the market. At such times, you rather invest, it takes 3 to 5 lakhs, but you rather buy the clone, that is, give a company the specific peptide and buy the clone. If you buy this clone, in future, the antibody is going to be very cheap for you. And it is your clone, so you can give it n number of times to any company. So think of these things before developing your point of care test. Now, luckily, because of this transfer of company, I would have the opportunity to go to these companies and get trained there. But rather train, they wanted the inventor to come there to duplicate the technology in their company. So I got to see what they do, and it's amazing. If you get training in a company, you come to know that they have so many tactics to reduce the cost even more. They have contacts where they can procure reagents for 1/4 of the cost which we are getting it for. And not only that, they have in-house tests, etc. For example, the COVID antibody kit, our working cost was 120 rupees. The company in front of me, showing what they were doing, they got it down to 70 rupees, which is almost half. Now, this is a beautiful thing, but at the same time, if they are big companies, at this point, they came to us saying, 3/4 we have done, it's only 1/4 you, so it's no longer an ICMR product. So over here, we face a lot of challenges. Try to get the cost of your POC. I know we are very happy when it comes to 100 rupees. Oh, it's cheap. Try to get it even lower than 100 rupees, but for some reason, the private companies can get it even more down. So if you don't want to lose on any of this or don't want to face problems, just try maximum to get the cost as low as possible. Tie-ups and collaborations with companies is beneficial. For example, I've never read this in a publication. This is something, again, I came to know only when I went to the company. So we standardized the kit using 60 microliters of plasma. It was there when they told me that nothing can be marketed or it's not usually commercialized if it is more than 30 microliters of your sample. This is something an academic would never. 60 is less. It is a very low sample volume. So what is 60 and what is their thinking is if it's a whole blood, you're going to prick yourself and the maximum number of drops would be 1 to 3. So how can you tell someone to squeeze in 6 drops? So that is why they say always standardize a kit with 10 to 30, that is 1 to 3 drops of the cell. So such things I think you get to know only once you collaborate with companies because they're doing that is their job. Now, when it comes to writing a project for funding, for technology, so there are many things which were said yesterday. All of this applies here also, even in the study objective or the aim. As ma'am yesterday said, you should be writing exactly what you want to do. For example, I'm going to develop a multiplex POC, which is going to detect XYZ things in this disorder, and it's going to be of a sandwich and competitive LSI. So this shows the reviewer committee that you know what you're doing and you've done your task of everything else, even in your methodology. If it is your first time, give 2-3 protocols of GMP synthesis. They know you're going to try the 3 things. So everything should be crisp and it should not be vague. Limitations of the study, what is it not going to detect? Gold standard detect. You should be very honest with this. The gold standard would detect this, but ours will only detect this. Be very clear. Timelines, as I said, is very important. Institutional support. Do you have those equipment, or the equipment can be asked for the funding? Now, expertise. Now, when you have the expertise, things become more smooth. If I say I've already developed 2, 3, they know, okay, maybe I can develop it. But if it's somewhere you need to start, it's always the first project for your first LFI. Over there, I would suggest do a little preliminary work. When I say that, order a few membranes. You don't have to order antibody or strip cutter or anything of that. Like I said, you can do it on dots. But know that you can make the GNPs. These are all very not that expensive things. But at least you will have the confidence. Yes, we have done something preliminary. We've tried to make gold nanoparticles. We've ordered the membrane to do that thing before submitting it for your funding. Now, coming to the budget, according to me, for any LFI could range from 25 to 55 lakhs. Generally, it doesn't go beyond that. And the main cost of this is your monoclonal, polyclonal antibodies, or the clones if you want to buy, the peptides and proteins. So there's one thing more which I want to say. So when you're developing something which has your sample is whole blood or urine, and it's a particular analyte, try to get a lyophilized form of that protein first. It is always better to standardize it with that protein itself and then go ahead with the blood. It becomes easier. So you need to take account of those peptides and proteins too. Very important, again, highlighting you need dedicated staff. I highlight this because many times what response you get is it's an excellent project, but do it intramurally. The point of intramurally is that you have your staff doing 3 other work. You cannot develop a technique when they're doing 3 other tasks along with. And equipment, it's a one-time investment. So your first project takes care of the line cutter and line strip cutter. After that, you never have to ask for it again. So it's a one-time investment. So now, to conclude, all the points to consider while developing a point of care kit. As I said, is it really necessary? The need of it. If it is necessary, please stick to the assured criteria. You need to have gold standard tests along with it. Otherwise, there's no point. If you do not have the gold standard test, you collaborate with an institute who is going to the gold standard test. Should be developed according to who will be doing it. Is it a lay person, or is it in the hospitals or the caregivers or something? Diagnosis and treatment has to go hand in hand. Yes, as I said, if you are making a patentable technology, excellent. If you're not, make in India product also makes complete sense. So please go ahead and do that, as long as it is cheaper than what is out there and more sensitive and specific. Principle, whatever technique. Now, I have highlighted LFI. You know that there are n number of point of cares there. Know your principle, know it thoroughly before you start your technology development. Instruction sheet, keep it simple, keep it very simple and clear. You need to do an internal and external validation with precision. Without this, nothing is going to go anywhere. You need to do stability tests simultaneously, and you need to set a timeline. You need dedicated staff. You need to, here I have mentioned antibodies, but please develop a technology with at least 2 or 3 sets of reagents so that the blunder which happened to us doesn't happen to anybody else. Before commercialization and transfer, try reducing the cost to the maximum. And the last is collaborate with companies. This is not started, but this could be a very good thing for ICMR institutes. So I want to end on a lighter note. So since the topic is points to remember or to consider before developing, along with the scientific, I think a scientist or inventor should know the emotional turmoil which goes through while developing. So just on a fun note, at the start, you get an idea, you are very excited, I'm going to be the greatest person in the world for developing this kit, and you are super excited. It even increases your ego when you get it funded. You get the funding, you are excited, you start it. But once you start it, at least for a drag of 4 to 5 months, you're getting no results. This is a point where you feel like, I don't know, banging your head to a door or something like that because there are no results. To add to this, your director or ICMR headquarters keep calling weekly, asking what are the results, what are the results. So it's like frustration with fear, with everything coming to this thing. But do not give up. You need to troubleshoot all your problems. You need to try, because trust me, after the 6 months or 4 months of breaking your head, it is successful. You do get successful results. At this point, again, you think you're the best person in the world and you need an award or something like that. And you give parties to everyone who you meet kind of thing. But it does not end over there. It is for the first time. You realize it's only the first time. It is after the technology is transferred. Also, there are many issues with rise up. So do not party until it actually comes up in the market. So, thanks.
I have a couple of comments that, once you're developing so the technology is developing, so as an inventor, so you rely on the, commercial intelligence.
Yes. Yes.
So, would that component come into the idea? So those are not actually developed by you,
Yes.
but are, so do we need to contact those companies, for example? What
Yes, yes, Sigma, for example, any information.
covering technology, your initial.
So the patenting happens based not on the reagents which are used, but on the technology. So that is why nowhere we have patented it, and nowhere they have asked us to get in contact with the companies. But yes, we do contact the companies because we know that we'll be needing them. So they don't make us write it anywhere, but they actually provide it, and they tell us which company has got it. We've provided them at a better rate. So till date, ICMR meaning we have not had such a situation where we have to mention it. But yes, your protocol when you're technology transferring, it actually says this antibody is from this company and this catalog number. So that you have to.
And also, like, there's a buzzword for any, and this sample has, so, and when I interact with the thing, I want to test, like, pregnancy to do. And pregnancy is what, hormone release, actually, you see the beta, that is very, common, you can make a so you can but when it comes to developing cancer, the question is, do we need a time because, in cancer, it's a screen and treat, right? So you have diagnosed one test, maybe an hour or a day or two. So you have the technology, which can detect in maybe half an hour.
Okay.
So does it really, translate into, it can be called it as a point of care? You have detected a disease half an hour compared to two, but you do not have, treatment strategy, which you can take a decision. So how do you cope up with those kind of things? You, instead of have your gold standard is able to detect in 2 hours, but yours is detected in maybe 2 minutes. How does it help a patient in terms of, point of care? Would you call that a point of care?
See, the point of care is anything which gives you results within 30 minutes. So if you're saying, and it is portable, it doesn't need a laboratory facility or something like that. So this would come, but you're asking me whether, meaning you're saying after detecting this also, the treatment is not, so there I wouldn't have an answer, but then why the gold standard? The gold standard tests are there and you're still doing that.
So that's why I follow up this. If I have a gold standard already, which can give me a test for working in cancer.
It gives you
So I know that
information.
cancer for the line of treatment.
Correct.
And it will take a time.
Correct.
So my thing is, can I call it as a point of care because I'm just reducing the time of detection.
Correct.
But there is no benefit to the patient actually in terms of treatment.
So no, that is why I said the diagnosis and treatment has to go hand in hand. So I think this would be a simpler test. That is what you would call it, but not a point of care.
Also, if you have a gold standard, for example, for example, you said it costs 35,000 or 35,000. And within that gold standard, if I make an innovation, that okay, I follow everything in gold standard, but I have something which I can reduce.
Yeah.
The
cost.
The cost. Can that be as a percentage?
So we consider that something, but that doesn't come to point of care test. It's simplifying the test, and if it is
because
it has to have different components.
You are following rather than developing a new test. You have already gold standard, but your target is make it cheaper.
Yes.
But you are not compromising those, maybe time effective or cost effective. So is that patentable?
It is. So then when you contact the patent office headquarters, they say you need to give the difference. What is different from that? And if it's just the cost, it's not going to be patent. there has to be some process also in that. So basically that all has to take care. So there are many times we made a simple test that we've given the entire protocol to headquarters. They give it to their legal teams and we've got an answer, this is not patent. We've again nudged them and said, but this is different, this is different. But when it's just reagents and reducing the quantity or something like that, it becomes cheaper. But that's
I have a question, ma'am. If you are doing LFA, so how you are going to choose your antigen? Anyhow, antibodies can be synthesized, of course, but what about the antigen?
The antigen and antibody. So basically, you should know the principle of the test, and both the antigen and antibody, depending if it's a sandwich ELISA, you would want the antigen too. So you get them in the market. The main goal of LFI
No, ma'am. My question is how you choose which antigen, target antigen in the clinical samples. How we have to see which antigen you are targeting, like that.
So that totally depends on what are you now. Suppose, for example, it's hemophilia or bleeding disorder. My antigen would be factor VIII.
If it is a bacterial pathogen, ma'am, say for example, chlamydia.
So the most, the epitope on it, which is the most, has the highest affinity, I think that you would choose. So basically, the more easier way is if you have a gold standard technique, what is the antigen they are targeting? That would be the most better way to choose your antigen.
Okay. Is there any method to screen the most expressed antigen in the particular bacterium?
Yes, so not only antigen, even antibody. Generally for LFI, there is one before that, you do an ELISA. So then in that ELISA, you use your N number of antigens, whichever is there in the market, or antibodies, and you see which has the highest affinity. Those ones you can take up for LFI. But basically, majority of the POCs, since they are in mission mode, we skip the ELISA step and we go directly and trying to standardize it on the LFI. But yes, that could be done if you want to know the best antigen.
Anywhere, ma'am, we can get trained with for this LFA.
Yeah. So, your experience as an Indian, once you have an idea and you're submitting your technology to maybe for ICMR, they have their own mechanism. By the time it takes years, sometimes months, years, and you realize that somebody else, maybe how the confidentiality is moving. But for example, you submitted your technology to me and anything for review. Yeah. And how that process, how do you
but I don't think so.
We are not able to hear whatever the gentleman is speaking. Can you speak into the microphone?
Am I audible, sir?
Yeah, can you please speak into the microphone? Yes. We are not able to. I mean, it's not very clear what you are saying. We can't hear.
So my question is like, how do you maintain a confidentiality while you are submitting a technology for review? And over a period, because it takes time. You have to take legal opinion. How, what should be the constitution of, sort of.
So for a conference, first of all, we are not allowed to present it in any conference or leak any information. So till date, whatever we have patented, we have never shown it outside that we are doing this project. Not only that, in your SAT meetings, you have to keep it very light. We are developing it and that's it. So you need to have your own reserve zones of what you are going to show, because you can't show it all. And then when it comes to patent, I think that is very reliable. Here in ICMR headquarters, only that team has it and they give it to the precise ICMR. So I've never faced any leaking problem. It's from the inventor itself, because sometimes you get so excited, you want to present it in a conference, so you want to tell people that you've done this. That is where, so you need to have that. Otherwise, there's no leakage.
Actually, we are going to have a session speaking on IPR and other issues. Maybe these copyright can get in the so here we can limit to
yeah, excellent.
Then one question. You are depending on the companies for the molecular entity. Because companies provide you antibodies in higher cost, and another question is that any company can buy that antibody and develop that test. Because let's say it is a technique.
Yes.
And antibody is available in the market. Any company can go further development of that test. You can't control on that.
No.
So,
But even if your antibody is not from the company, your own clone, similar, someone else can do that. So if you're talking of cost, okay, now in an academic institute, if I order an antibody, it's going to be costly. But when we speak to that company and the company which takes up your technology, they buy it in bulk. And when they buy it in bulk, it is like almost 1/6 the cost. So there, if you're talking of cost, yes, you can make your own, you can do the clone thing, which I said, have your own the clone, it becomes cheaper. But that n number of other companies also are doing. So that does not negate your competition. Everybody is competing. But the clone thing would take time. So what am I saying is, see the antibodies which you are ordering, see that they are cheap, meaning we don't take an 85,000 for 1 mg or something like that. We take the
Expecting me to speak.
going to reduce to 1/6 when we do the bulk order. This we know for a fact.
Okay,
I have to keep the entire thing. Okay.
I think we're running out of time. So if there are any more questions, people write to Dr. Sumit and we will take it up with Dr. You will get a